Good morning, everyone. Let's wait for 1 minute so we can start. Today's lecture. Okay. Okay, good morning, everyone. Let's get started. Let me briefly review what we have covered on Tuesday, which are very important because this will be a very important point, and you're going to see problems on these topics in the final exam. And also, next week, you're going to see some questions about this in your quiz. So on Tuesday, we have learned two forms of the inversions, right? One is the paracentric, the other one is the pericentric. So these two forms of inversion heterozygous depend on the location where the inversion occurs. So if, for example, if the inversion does not involve the centromere, so this is the para, means the inversion is standing aside the centromere. However, if the inversion involves the centromere, including the centromere, then this is the pericentric. Then we learn how this kind of inversion event can affect the inheritance patterns, right? So because of the inversion, so we call the inversion heterozygous. What does that mean? That means like in an individual, if one chromosome is a normal one, so suppose the 5 genes are arranged in the right order, standard order. However, if the other chromosome, the homologous chromosome, is a inversion mutant, so as you can see here, so the BCD has been inversed. So if in this heterozygous, inversion heterozygous, so during the meiosis, these two homologous chromosomes cannot be paired in a standard way. It has to be different, right? For example, if these two chromosomes need to be paired together, so this requires one of them to be twisted like this, such that they can be paired together. So pair means like the DNA region with the similar sequence will be will be paired together, right? So like this, right? Okay, so then we have learned how. So for example, if this is a pericentric means the inversion involves centromere. So this form is a little bit easier. So we learned how what would be the resultant gametes if a crossover occurs between within this inversion region. So if there's a crossover occurring within this inversion region, so what would be the consequence, right? So so after this pairing, and after the crossover being occurred, so we still following the standard genetic analysis, we know we could have like 4 types of gametes here, right? However, if you look very close to these 4 possible gametes, some of them cannot survive because of what? Because of the unbalanced genetic material. So because if a gamete only has this chromosome, as you can see, the gene D is missing. So probably gene D is a very critical gene in this organism. So if a gene D is missing, so probably this will be a fatal situation. And also, you see 2 copies of A, right? So if too many gene product from A gene, so that might be causing a problem as well. So that's why. So so gamete like this cannot survive, and the same for this one, right? So only half of them can survive. The parental type can survive. Erase this. All right. Okay. So so I hope to point out this point for the pericentric inversion problem. So the gametes that cannot survive usually carry both duplication and the deletion, right? So as you can see here, if this is a pericentric for the inviable gametes, you're going to see both the deletion and the duplication. So for example, here for this one, duplication and deletion is D, right? D is deleted. For this one, D is duplication, but A is deleted. So this is one feature for the pericentric inversion example. Okay, so we also mentioned because of this inversion, and some gametes involving the recombinants cannot survive. So if you use this model to do the genetic mapping to analyze the distance between B and C or A and D, then this will cause problem, right? Because some recombinant progeny cannot survive. So so you're gonna see less recombinant progeny than expected, than what we expect to see. So this will this will underestimate the genetic distance between B and C or A and D. So I hope this is clear. And the same thing similar thing for the paracentric inversion case. So in this case, the inversion is not involving the centromere, but just on one side of the centromere. And if there's a crossover occurring between these 2 homologous chromosomes within this inversion region, then it is very complicated, right? So we have learned this last time. And so after the genetic dissection, we know those are 4 possible gametes produced from this paracentric inversion example. So half of them are the parental type, but in the middle, these two types of gametes in the middle will not survive because of what? Because of the deletion. So in the previous case, the peri. So for the inviable gametes, we see both deletion and duplication, right? But if this is a paracentric inversion for the inviable gametes, we only see what? Deletions. So here we see something has been deleted, right? So probably those genes are very important to the organism. So deletion will cause a lot of problem, might be fatal. So that's why gametes like this cannot survive. All right. So the same thing here. So no matter which type of inversion, either the peri or para, the gametes involving the recombinant will not survive. So that means like you're gonna. So if you use this model to do the genetic mapping, if you want to measure the genetic distance between those genes, if the crossover occurs within the inversion region, then you're gonna lose some recombinant progeny because they either carry the deletion or deletion and the duplication, then they cannot survive. If you lose some progeny with the recombinant, then you're gonna underestimate the genetic distance between these two genes, right? So this is example. This is important because in the final, you may see some questions like this. Right. So suppose this is a inversion heterozygous. So this is a standard, the normal chromosome, but this one is a inversion chromosome, and at this locus, so the standard chromosome has two recessive alleles for this gene recessive and this gene recessive, but for the inversion chromosome, so here we have a wild type dominant and another wild type dominant for this gene. However, so for this dominant chromosome, so this region has been inversed compared to the normal. Okay, so let's see what happened. So if so this is a dihybrid cross, right? So we can use this to measure. So in a standard way, we use this dihybrid cross, test cross. We can calculate the distance between these two genes, right? Two loci. However, if there's an inversion like this, so let's see what happens. Right. So there are two possible ways to have the crossover. So either the crossover occurs in this region outside the inversion, then it's okay. So if crossover occurs here outside the inversion, all the resultant gametes will survive, will be viable, right? So that's why. So this crossover or the recombinant progeny from this crossover will survive and it will be observable, right? So I hope you still remember. So this is very important. We learned this before, and the final exam will be a cumulative, right? So don't forget about the previous knowledge. So when we calculate the recombination frequency to calculate the genetic distance between these two genes, we usually conduct a test cross, and in the numerator, this is the number of the recombinant, right? Recombinant progeny, and the denominator should be the total number of the progeny, right? All right. Okay. Now, if the crossover occurs here outside the inversion, so then all the progeny from this crossover will be viable, so they can survive. The same here. However, we have learned this: if there's a crossover occurring within this inversion region, so any crossover within this inversion region, they are not observable. Why? Because if there's a crossover occurring here, then half of the progeny will die, right? Will not survive. Half of the gametes, because of the deletion or both deletion and the duplication. So we have learned this before last time. So if the crossover occurring within the inversion, then only the parental type can survive, and only half of the progeny can survive. The other half cannot. So we're gonna lose. So the recombinants product gametes or progeny from these crossovers will be lost. That means like this number will become smaller because some of the recombinants due to the crossover within the inversion region will be lost. So then, so if this number is low, the observed number for the recombinant progeny is lower than what it should be. If this number, observed the number of the recombinant progeny is lower than what it's supposed to be, then we're gonna see the the recombination frequency, right? So the result is like, so we're going to underestimate the distance between these two genes. So in this example, suppose if there's no inversion in the wild type, if these two genes are on the same chromosome and the map distance between these two genes is 10 map unit, 10 centimorgan. However, if we do a test cross like this, and one chromosome has an inversion, and if the inversion covers 5 map unit long, then if you have a situation like this, and if you do the test cross and use this formula to calculate the genetic distance between these two genes, so this region is totally gone. So you cannot observe any recombinant resulting from this crossing over in this region. So what you can get is, okay, you only get this part and this part. So if this is the situation, so although the real map distance between these two genes is 10 map unit, but you can only get 5 map unit from your experiment, okay? So this is the genetic, this is the influence, effect that the inversion heterozygous can affect the genetic mapping. All right. Does that make sense to you? I hope everybody can understand the biology behind. Okay. All right, so this is an example. I think this is an example problem from our textbook, and so this is the same situation. So, for example, the two genes are known to have a genetic distance of like 45 map unit between them. However, if you do a test cross, if we perform a test cross, a hybrid test cross to measure the genetic distance experimentally. So this is the data. So if you, everyone must be an expert in this, right? So we easily can calculate based on this data. We can easily calculate the distance, genetic distance between these two genes. If you use data like this to calculate, this only give you like 2% of the recombinant. So this is totally like underestimate, right? So suppose these two genes are that far apart, 45 mapped units. So we're supposed to see like 45% of the recombinant from this test cross. However, in reality, we only see 2%. Why? So one possible mechanism is, so in this dihybrid individual, so one chromosome has an inversion, and the inversion is quite large, right? Because so 45% versus 2%, so which means like, yeah, that's about like 43% of the 43% of the map unit is covered by this inversion. All right, so let's continue. All right, okay, so I think I'm done with this part. So this slide actually just summarize what we have learned for the inversion. So again, we have two forms of the inversion, right? The peri and the para. So make sure you understand the definition difference between these two. And so the common thing between these two forms of inversion is both inversions will reduce the fertility when the heterozygous for inversion, right? So we have learned this before, because if a crossover occurs between the two homologous chromosomes within the inversion region, then half of the gametes, or half of or the recombinant gamete will not survive. So they are not inviable. So this is the problem. And so what's the consequence? So what's the effect of the inversion on the genetics? So we know this will reduce the number of the recombinant progeny in the test cross, right? And so the immediate consequence is like so if there exists the inversion heterozygous, this, and if we use this model to do the genetic mapping, we're going to underestimate the genetic distance between loci. So, and the good thing for the inversion is it can, so within a population, so this inversion probably can increase the genetic variability. So, yeah, good thing or sometimes bad thing. Okay. All right. So I think the last major thing for the large segment of the chromosome mutation. So before we move into the translocation, so any questions about the inversion that I can help answer? Okay, very good. All right. Oh, by the way, so yesterday I received an email from evaluation department saying, so the online evaluation for this course is open. So if you have time, please go to the online evaluation website to evaluate this course, and please let me know what is good and so I should keep for the future class, and what in what point so I can improve to do better in the future class. Thank you very much. And okay, now, so another very important concept for the chromosomal mutation is called the translocation. So translocation, so this cartoon actually shows definition of the translocation. Translocation means the DNA exchange between different chromosomes. All right, so we know crossing over usually occurs between the same chromosomes, the homologous chromosomes. That's just the allele exchange, right? But if the DNA exchange involves two different chromosomes, so we use different color to represent the two different chromosomes. As you can see, if the purple chromosome and the yellow chromosome, if there's a DNA exchange like this, so the DNA content has been swapped between these two chromosomes, then this event, genetic event, is named translocation. Right? All right, translocation. So if there's a translocation between these two chromosomes, okay. So let's see. So if an individual like this, so if this one, so let's see how the translocation will affect the inheritance pattern, right? If an individual like this has a translocation, so it can produce two possible gametes. So one possible gamete is like, okay, the normal chromosome. So this is one possible gamete. So two normal chromosomes are coming together. So this is okay. This is normal gametes, and it can survive. It can be viable, right? And if two translocation chromosomes come together to form a gamete, this is still okay. Why? Because every gene has been so this is balanced because this gamete has everything in it, just like a normal gamete, right? Everything. Even there's a translocation exchange, but still like everything is there. So this is a balanced gamete, and this gamete can survive, and it is so therefore this gamete is viable. However, if we look at here, so this parent can also produce gamete like this. So if we look at these two gametes, you're going to see these two gametes do not contain the balanced gamete, balanced genetic material. So these two gametes are called the unbalanced gametes. So look at here. So something is missing, right? This part is missing, but something has been duplicated. So this part has been duplicated, but we're missing this part for this gametes. So if you look at this gametes, all right, so this part has been duplicated. Actually, let's see which part. Yes, yeah, this part has been duplicated. Yes, but the blue part has been missing. So this part has been missing. So these two gametes are unbalanced gametes because some important genes may be missing, and some genes may be maybe too many there, too many copies there. This can cause problem, and it might be fatal. So, all right. So this is the genetic effect that translocation may have for the inheritance pattern. All right. So let me. So we have learned this before. So so this diagram shows clearly how the translocation occurs, right? Okay. So first we have two normal chromosomes, and if there's a breakage on each of these two chromosomes, but on for some reason, if when they try to link together, so this end linked with this chromosome and this end of the first chromosome will be linked to this end of the second chromosome, then you're going to see the reciprocal translocation, and this is called the translocation. Right. Okay. So so again, so translocation. So if for example, in an individual, if there's a translocation occurring, so what would be the consequence? So what would the translocation event will affect the inheritance pattern? So this is what we want to learn. Okay, so this is a translocation parent, right? We see. So initially, so these two genes A, B are actually in a standard situation. These two genes should be located on different chromosomes. They are unlinked. However, because of the translocation, and this wild type A allele has been translocated to here, and on the same chromosome with the B, right? For this parent, because of the heterozygous, still like A wild type allele and recessive allele, and wild type allele. So this one is a wild type, right? Okay, now if we test cross this one with the double homozygous recessive, so this is a classical dihaploid test cross, right? Okay, now so two possible progeny can be viable. So first, viable progeny. Why do I say this? Because those are the two possible viable progeny, right? So, for example, so this two translocation chromosomes come together, then this gamete will be a balanced one, right? Otherwise, if one is a translocation chromosome, the other one is a standard normal chromosome, then that one is not balanced, so it cannot survive. So that's why, so if in this situation, if we look at the viable progeny, so this is one, and this is two. All right. So what do we see is like we always see parental type, right? Either the mutant type or the wild type, and the wild type always so these two dominant wild type alleles are always come together because of the because they are on the same chromosome due to the translocation, right? So this translocation will produce a genetic phenomenon called the pseudo linkage. Initially they are not linked. However, because of the translocation, yeah, these two alleles, wild type alleles are on the same chromosome. So if you do the tested cross, so this will give you a pseudo linkage. Right? So this is one possible genetic phenomenon you can observe because of the translocation. Right. Okay, question. Let me see. So yes, right. The problem here is, oops, sorry. The problem is here is, oops, where's my point? Okay, so so initially, if there's no low translocation, A and B are on this on a different chromosome, right? They're unlinked. So based on the second Mendelian law, these two genes will be sorted independently, right? So if there's no translocation, you're gonna see what? You're gonna see 4 types of progeny from this test cross, right? Either parental type or recombinant, right? So, okay, now after the translocation, so still, so if everything can survive, I mean, so if we do not worry about the balanced gametes or unbalanced gametes, you still, from this test cross, you still have this kind of like 4 types, because there could be like crossover between here or between here, right? So it can generate the 4 types of gametes here. However, because of the unbalanced situation, so like this. So if you do this test cross, you can only observe the viable progeny, right? Which means this is impossible to survive. This one is impossible to survive. You only see this. You only see this. So if you do a test cross, dihybrid test cross, and you only see the parental type progeny appeared in the next generation, what is the conclusion? The conclusion is, okay, the conclusion you can make is like these 2 genes are always inherited together, right? That means they are linked. So you're gonna conclude, okay, they are tightly linked, pseudo linked. So this is the biology behind. I hope, yeah, this is clear. Did I answer your question? Okay. Okay, you're very welcome. Okay, so let's see. This is a little bit complicated. So when we have a reciprocal translocation. So for example, the normal standard chromosome is like this. This is normal 1, normal 2, 2 chromosomes, right? And if there's a reciprocal translocation between these two chromosomes, you see the color change, right? And this will be the resultant translocation. So we say T1 means the translocation chromosome 1, and this is the translocation chromosome 2, so T2. All right, so if we have a progeny like, if we have a parent like this, so this is a translocation heterozygous, and then during the meiosis, these 4 chromosomes will be paired together like this. So this is the pairing configuration. So don't be surprised because of the. So this is not this doesn't look like the standard form, right? Standard, if there's no translocation occurring, so we're gonna see this 2 chromosomes are separately paired together, right? the N1 will be paired with another N1, and N2 will be paired with another N2 because they are homologous chromosomes. However, if for this translocation heterozygous, so the DNA with the similar region will be paired together. So that's why you're going to see this very strange but interesting form structure. So this is the pairing configuration for this heterozygous translocation heterozygous. So you're going to see the same region will be paired together like this, right? I hope this makes sense to you. All right. Okay. Okay, now, all right. So after the pairing, so there's a segregation, right? So the diploid cell is gonna go through 2 rounds of the division to form gamete. So okay, so if for the translocation heterozygous, if the pairing configuration is like this, so there are 2 possible ways for this 4 chromosome to be separate. The first one is the adjacent one, called the adjacent one, which up and down. So which means, so the segregation will occur like this. So I'm going to draw a horizontal line to separate this. So if the T1 and the normal one is going together, is coming together to form a gamete. And on the other hand, if the normal one and the T2, the normal one and the translocation 2, they come together to form a gamete. So that is called the adjacent one segregation pattern, up and down. And as you can see, so if one normal chromosome is coming together with a translocation chromosome, you're going to see what? You're going to see the duplication of the purple and the deletion of the orange translocation segment, right? So they are unbalanced. This gamete is unbalanced. The same for this one. So if a normal one come together with the translocation 2, so you're going to see the duplicated region as well as the deleted region. So this kind of segregation will produce inviable gametes. So that means like you're not going to see the progeny like this, right? Only if this structure go through the alternative segregation pattern. Let me erase this. So only if what is going up, so the T1 and T2, so the 2 translocation chromosome come together. So if this one come together with this one, so this 2 come together. And if the normal 2 and the normal 1, yeah, come together to form a gamete, and then those are balanced, right? Those are balanced gamete, and both situation gamete will be complete and balanced, so they are viable. So I hope this is clear and it makes sense to everyone. And definitely in the final exam, I'm going to give a question, multiple choice question like this to ask you. So what will be the resultant gametes, the viable gametes from this translocation heterozygous situation? Okay. So the key point is to look for the gametes that are balanced. So they have everything and no extra, no deletion. If you see the deletion and or the duplicated genes, DNAs, then that's a problem. So that gamete is possible, but it's usually is not viable. And the key point for this is this. Okay, all right. So this is the very basic knowledge of the translocation. Now let's see the application of this. So we try to learn the linkage, the association between this translocation problem with some human disease. All right. Okay, so we have learned the Down syndrome, right? Down syndrome. So before the 21 plus, we have 3 copies of 21 chromosome number 21 in human. So first, in the past, we learned the first possible mechanism that causes Down syndrome. So what is the first mechanism? So I hope you still remember non-disjunction, right? The non-disjunction. So yes, very good. Thank you. Yes, non-disjunction. Yes. So this is very important. I hope you please do not forget about this. Yeah, because in the final I may come back to this point to give you a question about this. Yeah, in the final. So yeah, very good. All right. So non-disjunction. So this is one possible way to cause this human disorder, the Down syndrome. Because of the non-disjunction, you're going to see probably the chromosome number 21 has like 3 copies, right? All right. Now today we're going to learn the second mechanism. So the translocation can also cause this problem. All right. So we have so this is a standard normal human. So in this human, in this person, we have 2 copies of the chromosome number 1, right? 2 copies of the chromosome number 14, all right? However, if there's a breakage on the chromosome 21, and there's another breakage on the chromosome 14, and if there's a translocation, so everyone knows what the translocation means, right? So there's a DNA exchange between the 21 and 14. Now we're gonna see this. And because look at this little thing. So this is the translocated chromosome. And because this translocated chromosome is very small, so there's almost no useful gene, not useful gene. There is almost no critical genes on this small chromosome. So this one actually can be lost. All right, if you look at the remaining 3 chromosomes, so they are one normal, right? Another normal, and there's a very long translocation chromosome. So these 3 chromosomes actually have almost all the required genes on that with a balanced DNA content. So this can be, so this one is a normal, actually. This one is still normal. You do not see any symptom if the individual has genetic content like this, even they lost this small chromosome, okay, because there's no critical genes on this small chromosome, translocation chromosome. All right, so if an individual has a genetic setting like this, so this is named the Robsonian translocation carrier. So this is a carrier. Carrier means there's a potential, but this one is a normal subject, but it has potential to have progeny, to have kids with the problem. So that's why we call this this parent as a Robsonian translocation carrier. All right. So if this carrier, this parent is going to produce the gametes, so during the meiosis, this is the meiotic pairing for these 3 chromosomes. Now we only have 3 chromosomes, right? We have 2 normal chromosomes and we have a very long translocation chromosomes. So because of this very unique situation, during the meiosis, these 3 chromosomes are going to be paired in this way, right? I hope this is clear. Okay. Now let's look at this parent. So this is the parent, right? This is the parent, the carrier, the Robsonian the translocation carrier. All right. So if this parent is married with a normal parent, and for the normal parent, yeah, it can produce the normal gametes, right? So we see 2 standard normal chromosome 21 and chromosome 14. However, so this is a Punnett square. I hope you can see this is actually a Punnett square. So on this side, this is the normal parent, but on this side, this is the carrier parent. So let's see how many possible gametes that this carrier parent can produce. Right? Okay, so first, if the normal one, the normal 21 comes together with the translocation chromosome. So on the other hand, the other gamete will only have the chromosome 14, right? All right, so this is one possibility. All right, so let's look at here. So if we have a gamete like this, produced by the carrier parent, and if so this gamete joined together with the normal gamete to form this zygote. So what is this? So as you can see, for the 14, chromosome 14, yeah, it's balanced, right? It's balanced because we just have the standard copy of the genes on the chromosome 14. However, for the chromosome 21, we have almost 3 copies. So this is equivalent to the to what we have learned before. So if we if due to the non-disjunction, if an individual has like 3 copies of 21 chromosome, then that's a Down syndrome, right? So yeah, in this case, the same, because of this gametes come together with the normal gametes, then this individual will have almost like 3 copies of the 21 chromosome. So this is a Down syndrome. Let me erase this. Okay. All right. So let's look at the number 2 situation. So this is Down syndrome. So it's clear because we have like 3 copies, right? 3 copies of 21. But if we look at here, so for this gamete only has a normal chromosome 14, and if this gamete comes together with the normal, it can form an individual like this. So can you see the problem? Why this is lethal? Because yeah, it's not a balanced zygote, right? We have like complete balanced chromosome 14, 2 copies, but we only have 1 copy of 21. So this one cannot survive. All right. However, if we look at the other possibility, so if this parent, the carrier parent, produce a gamete like this, the 3 and the 4. So if the 2 normal chromosomes come together, on the same time, the translocation chromosome will come alone to form a gamete, right? Yes, it's usually more lethal to have. Depends. So depends. So I think so. If yeah, this is a good question. So usually, like both the duplication or deletion will be a problem, right? So depending on how important the genes has been duplicated or lost. So if you compare the shape of the chromosomes, the 22 chromosomes in the human, you're going to see 21 chromosomes is relatively shorter than others. That means like they have less genes on that chromosome, which could explain why the duplication of that will not cause a fatal problem, but definitely missing the entire chromosome will be a problem. So it depends. I cannot say there's a unique, there's a universal answer to your question, but yeah, depends on what genes has been duplicated or lost. So I hope I answered the question. In this case, yes, because the entire 21 chromosome is lost. So that's a fatal. So you do not see. So this is based on our observations. We never see an individual with 21 chromosome is missing can survive. But yeah, the Down syndrome is okay. It can survive. Of course, you will see a lot of syndrome associated with this genotype, right? Okay, now let's look at the second possibility for this carrier parent. It can produce gametes like 3 and 4. So if these 2 gametes come together with the normal what's the results? So the normal gamete 4 will produce the normal standard normal individual, right? So this is totally normal. And if the translocation chromosome come together with the standard gamete, it will produce exactly the same genotype like this parent. So this is another carrier. So this kid will have the same genetic setting with this kid's carrier parent. So is that clear? Any questions about this slide? Okay, good. Okay, so this is another example of the translocation in the Drosophila. Let me see the translocation. Yes, that's right. Yeah, that's right. Exactly. Very good. So, oops. Oh, sure. Yeah. So basically, basically when we try to make a when we try to tell if an individual can survive or not, or it can produce any viable progeny. So depending on this individual's genetic content, right? So for example, for this carrier parent, even this individual only has 3 chromosomes, 2 normal chromosomes and 1 translocation chromosomes. So this little thing has been lost. Even this individual only has 3 chromosomes, but if you look at the DNA content of these 3 chromosomes, so these 3 chromosomes almost have everything like the normal standard individual. That means like so it will produce the same amount of the gene product like a normal human. Then this individual itself is a normal, and it can also produce kids with the normal phenotypes if this happens, right? So this is the totally normal situation, and this is a carrier like this parent. However, if for this situation, so because of there's too much there's too many copies of the genes on the 21 chromosome, that this will cause problem, and we're going to see the Down syndrome for this individual. So the key point is to check the balance, the gene balance, the gene content. If for any individual, just check if the gene content for this individual is comparable to a normal standard human or individual. If it is yes, then yeah, this is a standard. Okay. Okay, let's look at the example in Drosophila. All right. Okay, so there are 2 new concepts here. So euchromatin and the heterochromatin. So this represents the different part of the chromosome. So euchromatin usually means like the unwrapped, the open area of a chromosome, and so the genes on this region and close to this region can be easily transcribed and expressed. However, for some part of the chromosome, if the chromosome is tightly packed, very tightly packed, and it's wrapped, and even there's genes inside or close to this region, so those genes may not be easily to be transcribed, expressed. So those are the 2 different regions. And in the Drosophila, so we know there's a gene. So here we use W. So this gene is actually X-linked. It is on the X chromosome, and it will determine the color of the eye of the Drosophila. And so, okay. So this is X chromosome, and this is another chromosome, the chromosome number 4. So both chromosome has the euchromatin and the heterochromatin, right? Okay. Okay. So now, so actually, the X chromosome containing the W genes can be translocated with the chromosome number 4, like this. All right, the results is like, all right, because if this gene is on the X chromosome, it is located in the euchromatin region, so it can be always expressed. However, if there's a translocation, so if this gene has been translocated to the chromosome number 4, and if it is very close to this heterochromatin region, so sometimes it cannot be expressed, so it will be silenced. And sometimes it can be expressed depending on how close it is, right? So, or depending on the cell environment, so some other factors in the cell. So this is the situation. Okay, now, all right. So this is a standard. Let me see. This is a. Oh, sorry. Let me erase this. Okay, so this is a standard. So suppose if this is this wild type allele is located on the X chromosome and it is in the euchromatin region, so it is always expressed the wild type. Then this is a wild type. So you're gonna see the wild type the eye color for the Drosophila. For example, if the wild type is like the red eyes, so you're gonna see the whole eye is red. However, if there's a translocation occurs, and if this wild type allele is very close to this heterochromatin region, so this means like, so if this is the eye of the Drosophila, so because this wild type allele is so close to this heterochromatin region, so sometimes it can be expressed, sometimes it cannot. So that's why you're gonna see, so for this eyes, in some cells, you're gonna see the wild type, the red color, but for some cells, you see the white color. This is the mutant allele. All right, so this phenomenon can be explained by the translocation event, right? Okay, so, all right, and this slide just give you some very general ideas. So translocation sometimes can involve multiple chromosomes. So because in here, I only give you a simple example showing like the translocation occurs between just 2 chromosomes, right? But in reality, so it is quite often that this translocation may involve like more than 2 chromosomes. So for example, for this one, so 21, number 1 or number 2, and 14. So maybe the translocation will occur between these 3 chromosomes. So if you are interested in this topic, so maybe you can, if you have time, so you can search for some literatures to dig in to study how this genetic event can occur, and what is the consequence, and how this event can be related to some human diseases, something like this. It's very interesting. Yeah, fun study. Okay, so this is another example in humans. So the leukemia, the adult leukemia. So actually, 15% of the adult leukemia is due to the chromosome translocation. So this involves 2 chromosomes, the chromosome number 9 and the chromosome 22. So if you see, for example, like this, so if the translocation between 9 and 22 occurs like this, this will form a hybrid oncogene like this, right? Okay, so I think I'm done with this this chapter. So before we move forward, so any questions for today, for this chapter? Okay, very good. And so let me just start talking about the last two chapters. I don't think we're gonna go too far. So the last part for our class, yeah, this quarter. So you're almost done, almost done. Congratulations. And so this part is so it's good, but it's not that important compared to the previous part because so in the last two lectures, I'm going to introduce some very interesting, useful concepts that will be helpful to your future career. But okay, yeah, yes, me too. I really hope like you enjoy this class. So usually I teach class. I hope like student can benefit, can learn from this, and hopefully some of this can be useful for your future career. Yeah. Okay. Thank you very much. It's very nice of you to say that. Yeah. Lots of puzzles. Yeah. That's the point. Because if you not feel like challenged or stressful in a class, I don't think that class will be useful to you. So usually, like, yeah, learning is a painful experience, but yeah, yeah, if you spend efforts and time on this, yeah, it will be paid back, and yeah, everybody grow in this way, right? Yeah, I really hope like this class, yeah, you enjoy this class and yeah, and learn something useful for your future career. Okay, so for the next two lectures, I'm gonna introduce some interesting, important concepts that in the future, currently is a very hot area, and in the future, probably some of you is gonna following your career in this region, so in this area, then will be that'd be great. So we're gonna talk about genome and the genomics. So when we talk about genome, we mean all the DNA material in one cell. So that's the genome, complete genome. All right. So so all the DNA on the chromosome, of course, first, right? So including the genes. So the protein coding genes. So some meaningful genes and some like some genes we cannot say they are not meaningful because sometimes they look like junk to us, but maybe not. So this is because we do not understand genetic content in that region, right? So some important region. So we need you in the future to decode them and to identify the function of those non-coding gene the DNA region. So intergenetic sequence between genes. So actually 99% of the genome contain the genes that without so far without meaningful information, but in the future probably you're gonna figure out the function of those. All right, a lot of them are repeats DNAs. So in eukaryotes, so each cell may have like 2 or 3 sets of chromosomes. So of course, the major two to three sets of genomes. So the major genome will be the nuclear genome, right? So within the nuclear, that's the major DNA content. For example, like in human, the 46, 23 pairs of the chromosome, those are the major genome. And some organelle, for example, the mitochondrial, so it has its own genome. So it's also very important. and that's the mitochondrial genome. And the plastid genome, so for example, in the plants, the chloroplast genome, so that's also very important. So in the future or in the exam, if I do not specify an assay genome, usually if I do not specify anything, if I say genome, usually this genome referred to the nuclear on the major chromosomes. And so this is the human genome. So we have 22 pairs of the autosome, and we have 1 pair of the sex chromosome, right? And so there are about like 3 billion base pairs in a haploid genome. That's a lot. And on our genome, one set of the complete genome, so there are about like 30 to 40,000 genes. And so the genomics, actually, the genomics means like the study of the genome, right? It's the study of the genome, including the large chromosomal segment containing many genes. So in our class, so we just finished some of this, right? So we studied the large chromosomal segment rearrangement disorder or something like. So this is important. So this is a very important part for genomics. And so to study genomics, for the genomics, the study of the genome, so the first thing is to sequence them. So first we need to get the sequence information, right? Otherwise so it's very hard for us to study the genome. So the initial phase of the genomics will be mapping, sequence and mapping the entire genome, either the genes, the protein coding genes, or the intergenic regions. So we need to sequence all of them. And then the second phase will be functional genomics. So once we got this complete sequence of a genome, so we can study the functions. We can find out the functions, informations about the DNA sequence. Okay. All right. Okay, so I think I'm gonna stop here. So any questions? I'm gonna continue this next time. So any questions so far? So if not, I'm gonna stop here today. And please, yeah, if you have time, yeah, go to the online evaluation website and give me some suggestions or encouragement. So if you think anything is good, so I'm gonna, yeah, please let me know. I'm gonna keep for the future class. Or if you think anything need to be improved, also please let me know. I'm gonna do better next. All right, thank you very much. So I'm gonna stop here. Thank you. Yeah, you too. You have a great weekend. All right. So tomorrow I will hold at my office hours. In case you have questions, so I'm gonna see you tomorrow, Friday afternoon. All right. Bye-bye.
Professor?
Yeah.
Are you going to be doing a final clinic for next week?
Oh, yes, sure. For
Okay.
next week. So yes, we have 2 weeks, right? Oh no, we have 1 week. Yes,
1 week.
I will do that. Yeah, of course.
All right. Thank you, Professor. Have a good one.
Yeah, you too. Thank you. Bye-bye. Yes, we will get, yeah, for final. Yes, of course. Yeah, I'm gonna get, so I'm gonna prepare for a practice test. And so as you can see from the syllabus, so for the last lecture, we will not teach anything new, but I'm gonna take that opportunity to go over the practice test with you on the final, on the last lecture. Yeah, in the last lecture. All right. Okay, you're very welcome. Okay, any questions? If not, okay, bye-bye. See you next time.